Welcome to the screencast for section 5.1, cell division and the cell cycle. One of the first things we're going to learn is that there's a basic difference between the way prokaryotic cells, like bacteria, divide, and eukaryotic cells, the cells in our bodies, the ones that have a nucleus. We're going to look at the major phases of the eukaryotic cell cycle. We're also going to talk a little bit about how it's controlled and a little bit about how cancer relates to the cell cycle. So cell division is the process in which one cell, which is normally called the parent cell, divides into two daughter cells. As we mentioned in prokaryotes, that's simpler than it is eukaryotes, and one of the main reasons is those eukaryotic cells have a nucleus. And in that nucleus, the DNA has to go through some processing before it can divide. That's the basic difference. And in this section, we're going to take a look at first the prokaryotic cell. The vast majority of prokaryotic cells divide by a process called binary fission. And let's take a look at number 1 and number 2. So each start out as a regular bacterial cell, but the green color here represents, the green color right here represents the original bacteria. Notice that in the process 1, the orange, the new growth, is occurring at the center, and then it will pinch off, creating two new cells. Let's go to number 2 now, though, and you can see that the growth occurs at the ends of the bacteria cell. So it's just two different ways that the new growth starts. But the overwhelming majority of prokaryotes, the overwhelming majority of bacteria cells will reproduce by binary fission. Let's take a look at this diagram, which has a little more detail, and this is binary fission, so this is a bacteria cell. Here are the chromosomes, which are tightly wound DNA, and notice in the second one, the cell elongates a little bit, and the word replicates. It replicates its DNA. So whenever you see the word replicate, that means the DNA is going to double itself. It's going to copy itself. Why does it have to copy itself? Because eventually it has to reside in these two new daughter cells. So whenever you see the word replication, all that means is duplication. It's copying itself. It's doubling itself. As we mentioned, the eukaryotic cell cycle is a lot more complicated, and this diagram, which will show up in this slide and in the next slide, I'd like you to really study and pay attention to. So let's say a cell has just divided. That's right down here, and it will go through the first step, which is called the first gap, G1, before it gets into this step right here, which is called synthesis. So the cell is growing and moving along here. All of this is a phase. See the dark green here? All of this is a phase called interphase. But in the synthesis part of interphase, that's where the DNA doubles. And then we go into the second gap, G2, and then this is where the nucleus doubles in stages called prophase, metaphase, anaphase, telophase. Cytokinesis, the cell actually divides. It's now two daughter cells, and the whole cycle starts again. We're going to see this in a little more detail in a second, but make sure you understand that this mitotic phase, this is mitosis, this is where the nucleus itself is dividing, and this is the interphase where the cell spends most of its life, and the synthesis phase is where DNA doubles. We'll take a look at this a little closer, and we'll show you some actual pictures of it. So in this slide, we go into a little more detail. We just basically take the diagram that we just saw. So let's go with point 1 here, cell growth checkpoint. This occurs at the end of G1, and it checks whether the cell is big enough and has made the proper proteins for the synthetic phase or the synthesis phase. So right here, proteins do all of that checking. Then number 2 is when the DNA synthesis checkpoint. If everything's good, DNA starts to copy itself. And then after DNA has copied itself, we go around here and we get to the beginning of the mitosis checkpoint. And that occurs in the mitotic phase in metaphase, and checks whether all the chromosomes are attached and ready to begin splitting. If so, the cell divides and the cycle repeats itself. So 1, 2, 3, these are the checkpoints in the cell cycle. So sometimes the cell cycle and the function of cells gets messed up. It gets destroyed. And in this case, the cells will start to grow very, very quickly. They will no longer function as the type of cell they were supposed to function at. For example, let's say a muscle cell can no longer contract and starts growing uncontrollably. That many times is called cancer. One of the reasons why cancer is so difficult and why it creates all the different types of tumors it does is because we have so many different types of cells. And any cell, practically any cell, can become, a cancer cell. Let's take a look at section 5.2. So we're combining in this screencast 5.2, and 5.2 is going to be short for us, but we're going to talk about chromosomes and mitosis. Now remember, chromosomes are nothing more than tightly wound DNA, and actually you can see chromosomes with a light microscope. We're going to see that in a few seconds. So our objectives here is to describe chromosomes and their role in mitosis and outline the phases. I included the vocabulary because there's lots of new vocabulary in this section that you may want to study and understand. So let's go to the next page, and this is a really nice stylized version of the cell cycle. So here's mitosis in the cell cycle. So here we are in G1. All this is interphase G2, but then something starts to happen down here. And in this case, the DNA starts to duplicate itself. The chromosomes duplicate itself. Instead of having 46 paired chromosomes, they're going to double and get ready to be in 2 daughter cells. So prophase starts that. Then when the chromosomes have all doubled, they all line up in metaphase. Then they are pulled apart in anaphase. Telophase, they're in 2 separate areas of the cell, and then they pinch off and they basically divide in cytokinesis into the 2 daughter cells. So let's actually take a look at what this looks like. Here is a frozen section of onion cell which has been stained. And I just want to point out 2 of the sections. So we're going to go back for a second. We're going to look at metaphase and anaphase. So let's look at what that looks like in real life. So right here, all the chromosomes are lined up in the middle of the cell. This is metaphase. Can we see another metaphase here? Yep, here's metaphase right here, just before they're pulled apart. Let's look at anaphase, where the chromosomes are being pulled apart. Here's anaphase right here. Here's another cell that's in anaphase here. This is probably prophase. This is probably just before the cells move to the pole. A lot of these other cells are in various stages of interphase. So these are onion cells which are stained blue, showing you the stages of mitosis in the cell cycle. So in the next slide, we're going to take a look at a cell where the chromosomes are all lined up right here in the middle of the cell in metaphase. And we're going to look at some specific parts. So these are chromosomes consisting of the sister chromatids. That's what each of these is called, the sister chromatid. The chromosome itself is this whole thing, and this one side is the sister chromatid. The centrioles are attached to those chromatids. They will pull them apart. And the centromere is the center of the chromosomes, or the center of the 2 chromatids. And these terms you should take a look at. These microtubules are sort of like the ropes or the strings or the cables that attach and pull it apart. It's an amazing process. Okay, that ends our screencast for section 5.1 and 5.2. Please make sure you read those sections, study them, take notes, and we'll be moving into screencast for 5.3 now.